Hi there, science fans. Welcome to another episode of Science and Other Stuff for Senior Citizens. My name is Dan Casciano. If you like these COVID-19 videos, please subscribe to my channel and ring the bell to be informed of future videos. In today's video, I will be describing some coronavirus biology as it relates to SARS-CoV-2, which is responsible for the COVID-19 disease. The description includes some molecular biology that hopefully is understandable to you. If you would like a more in-depth molecular description in future videos, please leave me a comment. This slide shows the structure of the SARS-CoV-2 virus. The outer surface of the virus is studded with structures called spike proteins. The arrangement of these structures resemble a crown when the virus is viewed through an electron microscope. Thus the prefix corona, which is Latin for crown, in the word coronavirus. There are also several proteins and a lipid membrane that make up the remainder of the outer virus coat. This outer shell surrounds and protects the RNA informational molecule that contains the blueprint instructions or manual for making another virus, as well as the spike proteins that are used for entry into the host cell. This RNA is also associated with nuclear proteins depicted in blue in this cartoon. Once inside the cell, the virus must hijack the host cellular machinery to make more viruses in order to infect other host cells and to infect other human hosts. Here we observe how the virus initially interacts with the target lung cells. The surfaces of many cells of the body contain protein molecules called receptors that are used for a variety of purposes, many of which have evolved to assist in the survival of the cell. Certain viruses and other pathogens have also evolved to use these receptors for their own purpose. In this case, the spike protein latches on to the ACE2 receptor located on the surface of lung cells. As you can see from this diagram, the interaction is very specific, like a lock and a key. This diagram shows a closer look at the cell's surface. The spike protein structure contains a stem segment that contains fusion machinery and the spike protein. The function of the protease is to act like a scissors and cut off the top of the spike, a requirement for the virus to gain entry into the lung cell. Once the spike is clipped off the surface lung protease enzyme, the spring-loaded stem fusion machinery unfolds to attach to the lung cell membrane, allowing fusion of the virus particle with the cell. This fusion results in the dissolution of the cell and virus membranes, allowing the injection of the virus RNA into the host lung cell. This process very much resembles the process by which a sperm's DNA is injected into a female's egg cell, resulting in fertilization. This slide describes the entire process by which the virus appropriates the lung cell protein-making machinery to make another virus. The RNA instructional molecule first makes several protein molecules needed to make a new RNA instructional molecule. This process is depicted on the left side of the slide and is represented by a cluster of bluish-purple colored proteins that becomes the replication transcription machinery. This structure is the place where more viral genomic RNA molecules are made, then are translated into viral proteins required for the assembly of new viral particles. After these particles are assembled, they move to the surface of the cell to be released to infect other cells or new hosts. All of this synthesis takes place at the host cell expense. The virus cannot survive on its own and requires a host's energetic and synthetic capacity to produce a new offspring. In previous videos, I described the human immune system that evolved to protect us from invasion and destruction by foreign pathogens like viruses and bacteria. Briefly, an innate and adaptive cellular immune system have evolved and are depicted here in this slide. The innate system is made up of a number of different immune cells that are nonspecific in nature and respond to any foreign invader. When a cell is infected by viruses, it makes several signaling proteins, one of which is called interferon. Interferon alerts the innate system to marshal a defense against the invader. In the diagram in the upper left quadrant, there is a cell infected by viruses. The cell is making the signal protein interferon presented in yellow that then recruits a variety of cells, one of which is a macrophage that recognizes the infected cell and proceeds to destroy it. The interferon also alerts cells of the adaptive immune system presented in the diagram on the right. B and T cells are a major component of the adaptive immune system. B cells are responsible for producing the specific antibodies used to neutralize the invading virus. The T cells differentiate to killer cells that kill infected cells and also differentiate or change into T memory cells. The T memory cell will recognize the specific virus and will mount a defense against it. This process is explained in more detail in previous videos. The virus is also an evolving biological entity that responds to these defense mechanisms by developing countermeasures to sustain its survival. In the left panel, this particular virus produces certain defensive spike protein surface molecules that trick the neutralizing antibodies, resulting in survival of the virus. In the middle panel, we see how a cell using sensor protein molecules recognizes the presence of a foreign invader and signals the cell's genome to make interferon proteins. A proposed viral countermeasure is depicted in the far right panel. Here, the sensor protein is inactivated by a viral protein blocking the synthesis of cellular interferon, resulting in the inability to recruit more cellular defense mechanisms. In addition to the development of vaccines to encourage the body to mount a robust immune defense from SARS-CoV-2, drugs are being developed to target specific areas of the viral infection pathway. These include drugs that prevent the virus from entering the cell, drugs that shut down the virus, drugs like remdesivir that encourage synthesis of defective viruses, and drugs that decrease a hyperimmune response such as dexamethasone. Here are sources I've used for this description. Dr. Britt Glausinger has several YouTube videos describing coronavirus biology, and much of the illustrations were developed by Veronica Falconieri-Hayes. Although a vaccine is on the way, we must continue to clean or wash our hands often, avoid close contact with sick people, practice distancing 6 to 10 feet, avoid indoor and outdoor crowds, and wear a mask. For more information about science, visit my website at www.dcasciano.com. If you have any questions or would like me to develop a video on a specific science subject, leave them in the comment section below or email me at dan@dcasciano.com. Be sure to like this video and subscribe to my channel and ring the bell so you can be informed when I upload a new video. Thank you for watching Science and Other Stuff for Senior Citizens. Bye.